Now let's discuss about the procedure for the initiation of callus culture from the taproot of carrot. First of all, a fresh taproot of carrot is taken and washed thoroughly under running tap water to remove all surface detritus. The taproot is then dipped into 5% tea pool for 10 minutes and then the root is washed. The carrot root, sterilized forceps, scalpels, and other instruments, autoclave nutrient medium petri dishes are then transferred to laminar airflow chamber. Throughout the manipulation sequence forceps, scalpel must be kept in 95% ethanol and flamed thoroughly before use. The taproot is surface sterilized by immersing in 70% ethanol for 60 seconds, followed by 20 to 25 minutes of sodium hypochlorite solution. The root is then washed 3 times with sterile distilled water to remove completely the traces of surface sterilizing agent. The carrot is then transferred to a sterilized petri dish containing a filter paper. A series of transverse slices 1 mm in thickness is cut from the taproot using sharp sterilized scalpel. Each piece is then transferred to the another sterile petri dish. Each piece contains a whitish circular ring of cambium around the pit. An area of 4 mm square across the cambium is cut from each piece so that each small piece contains part of the phloem, cambium and xylem. Size and thickness of the explant should be uniform. Always the lid of the petri dish is replaced after each manipulation. The closure, that is the cotton plug from a culture tube, is removed and flame the uppermost 20 mm of the open end. While holding the tube at an angle of 45 degrees, an explant is transferred using forceps onto the surface of the agarified nutrient medium. MS medium generally used for the culture. The closure is immediately placed on the open mouth of each tube. The forceps are always flamed before and after use. Date, medium and name of the plant are written on the culture tube by glass marking pen. Culture tube after inoculation are taken to the culture room where they are placed in the rack. Cultures are incubated in dark at 25 degrees centigrade. Usually after 4 weeks in culture, the explant incubated on medium with 2,4-D will form a substantial callus. The whole callus mass is taken out aseptically on a sterile petri dish and should be divided into 2 or 3 pieces. Each piece of callus tissue is then transferred to the tube containing fresh culture medium. The prolonged culture of carrot tissue produces large calluses.